All right, this is a screencast to introduce you to two-way analysis of variance with replication. There are two forms of analysis of variance in a two-way type of situation. One involves when you have replication of each of the two treatment combinations, and the other one involves a situation where you don't. And so we're going to go through with replication because it is most similar to what you did with a one-way ANOVA before. So just to give you an example of what this is like, if you go out on I-70 west of Kansas City and you get to the Manhattan area and look to the north side of I-70, you'll see a large research area there called Konza Prairie. And at Konza Prairie, ecologists are studying the effect of fire frequency and the effect of native grazers, i.e. bison, on the plant community there. And so they have, in this green section here, they have a bunch of watersheds that are divided up by watershed, and they have these numbers. This is in 1A, in 2B, in 4C, and in 20A. The numbers 1, 2, 4, and 20 refers to how many years they allow to pass before they burn this watershed. So in the 1 watersheds, they burn those every year. 2s get burned every 2 years. 4s get burned every 4 years. And 20s get burned every 20 years. But then also in this green area, they have bison running around. And the bison are free to roam wherever they want to within this. So over the entire course of this area, bison are occasionally grazing. But they've also created small plots within these watersheds that fence the bison out of areas. So within these larger watersheds that are getting grazed by bison, there are little plots that are not getting grazed by bison. So I always like to visualize things, and so I made myself a little graphic. Here we have two watershed types, watersheds burned every 1 year and watersheds burned every 4 years. Within these watersheds, you have bison grazing everywhere, but then also within these watersheds, you have little fenced off areas where the bison aren't allowed to go. So outside of these squares, bison are grazing. Inside of these squares, the bison are not grazing. But when you burn this watershed, both the area inside the enclosure and outside the enclosure all get burned. Another way of thinking about this is to think about it in a tabular form where we have one effect is grazing, another effect is burn frequency. For grazing, we have two treatment levels, ungrazed and grazed. And I like to do this once again with pictures. So in the grazed area, you have bison present. And then in the burn frequency situation, we have 1 year burn frequency and 4 year burn frequency. The 1 year burns are getting fire more frequently, and the 4 year burns are getting fire less frequently. So you can look at the effect of grazing, which is the columns, and you can look at the effect of burn frequency, which is the rows, independently of one another, but you can also look at how they interact because you have all combinations of both grazing and burn frequency. This is similar to the situation with the geckos, where you have summer and winter, and you have Galveston and Oklahoma City. And so you have two treatments, but you have all combinations of them. You have data from Oklahoma City, both for summer and winter, and you have data from Galveston for both summer and winter. And so you can look at each of those factors independently of one another, and you can also look at those as they interact with one another. So it's very similar to what we do with a single factor analysis of variance, in that we have to calculate the sums of squares, but doing the sums of squares, there's just more of them because we have now two treatments. In the case with the bison, we have row, which is burn frequency, and column, which is grazing. But in this case, we just take the average of the row across both grazing frequencies. So we take the average of 1 year burns across both grazed and ungrazed plots, and we take the average of 4 year burns across grazed and ungrazed plots. We get those row means, and we subtract the grand mean from the row mean and square it for both rows, and then we multiply it times the number of columns, which is Q, and the number of observations in columns, which is N. And that's how we get our weighting for the sums of squares for for, in this case, burn frequency. To get the sums of squares for grazing presence or not, we look at the average across all burn treatments for grazed minus the grand mean and ungrazed minus the grand mean. Both of those are squared. They're multiplied times the number of rows and the number of observations in each row. These are the degrees of freedom for row. You have p numbers of rows minus 1. For columns, you have q numbers of rows minus 1. The interaction term we're going to calculate indirectly, so let's just leave that for now. But we also then are going to take the residual. So the residual is the same thing as sums of square error or sum squares within. These are all interchangeable terms. How do we do that? Well, this is the within sample variation. So we take the individual values minus the sample mean squared. We do that for all four, in this case, all four treatment combinations, and add those up. And we don't have to do any weighting because we have the same number of observations in each of these things. We're adding those up across all the observations that we have. And then sums of squares total is just like we've done it in the past. It's each individual value compared to the grand mean squared. And once again, we don't have to weight this in any way because we're dealing with each of the data points individually. These are the degrees of freedom for the residual and the degrees of freedom for the total. So let's look at how this gets done when we look at an actual data set. So this is a tiny data set in terms of total size. There are 12 observations, but those 12 observations are divided up across fat type and across males and females. So this was an experiment. It's a textbook experiment once again, where researchers were looking at the effect of the type of fat that rats eat and whether that fat is rancid or whether that fat is fresh. Clearly, you might expect rats to eat fresh fat more than they would eat rancid fat because, of course, rancid fat is rancid and doesn't taste very good. So we're looking for an effect of fat type on weight gain, but there's also the possibility that males and females might behave differently across these two fat types, so we can divide our samples into males versus females. And for each treatment combination, we have 3 observations. We have 3 males that were fed fresh fat, 3 males that were fed rancid fat, 3 females that were fed fresh fat, and 3 females that were fed rancid fat. So to begin with, we need the grand mean. So we need to sum up all of the data and divide by the total number of observations that we have, and that gives us a grand mean of 597.75. We can then, now that we have the grand mean, we can calculate the sum of squares total, because sums of squares total is just each of these individual data points minus the grand mean squared added up across the entire data set. So it'd be 709 minus 597.75 squared plus 679 minus the grand mean squared plus 699 minus the grand mean squared plus 592 minus the grand mean squared, 538 minus the grand mean squared, 476 minus the grand mean squared, 657 minus the grand mean squared, 594 minus the grand mean squared, 677 minus the grand mean squared, etc., until you've done the whole thing. When you do that, you should get a sum of squares of total that should be 77,570.25. Take a moment, do those calculations either in an Excel worksheet or on a sheet of paper, and verify that you get the same sum squared total that I get. So hopefully you got the same answer that I got. So sums of squares row is going to look at differences between males and females. So the first thing you need to do is you need to calculate the average weight gain for all males regardless of fat type. So what we do is we take all 6 of these values that are values from the male, we average them, it should give you 615.5. We add up all the data for females, and that should give us 580. You see that males are gaining somewhat more weight than females are, probably because they probably started out heavier. because of sexual dimorphism, but the sums of squares row is due to 615 minus the grand mean squared plus 580 minus the grand mean squared, and then you take that whole thing and you multiply it times the number of columns, which is 2, and the number of observations within each column, which is 3 for males. So that would be that sum of 615 minus the grand mean squared plus 580 minus the grand mean squared. That would then be multiplied times 2 times 3, which is 6. And what you should get out of that is 3870.75. Once again, take a minute, work through that set of calculations and make sure that you're getting the same value that I am getting. If you're having trouble with this, go back and reference these equations to help you out. In a similar way, we can look at the effect of fat type by calculating the average across male and female of fat type. So this would be column means. 669.167 for fresh fat and 526.33 for rancid fat. You'll notice that these averages are much further away from 597.75, the grand mean, than the row means are. So you might expect this sums of squares to be larger. So if you take 699.16 minus the grand mean, square it, 526 minus the grand mean and square that, add those together and then weight it by, once again, 2 rows with 3 observations each, so multiply that times 6. What you end up getting is a very, very large sums of squares due to column, 61,204. So to calculate sums of squares within, we actually are looking at within sample variation, so we need the average of each of the treatment combinations. So that's what I have listed here as subgroup means. So we need the average of male fresh growth, which is 695.67, male rancid growth, 535.33, female fresh is 642, and female rancid is 517. And so these then are compared not to the grand mean, but these are compared to the individual values within that treatment combination. So this would be 709 minus 695.67 squared plus 679 minus 695.67 squared plus 699 minus 695.67 squared plus 592 minus the sample mean, 538 minus the sample mean squared, 476 minus the sample mean squared, 675 minus the sample mean squared, 594 minus the sample mean squared, 677 minus the sample mean squared, and then do the same thing for female and rancid. What you should get is 4 different sums of squares within for each of the treatment combinations. 466 corresponds to fresh male, 6738 male rancid, 3752 female fresh, 708 female rancid, and that all adds up to 1166.67. Now the easiest way to find the row by column interaction sums of squares is by taking the total sums of squares, subtracting from that the sums of squares due to row, sums of squares due to column, and sums of squares within. And when you do that, it'd be 77,570 minus 3780 minus 61,204 minus 11,666. That should leave you with just 918.75. So take a minute, do all those calculations, and verify that you're getting the same answers as me. Just pause the video. Okay, now we can take these values and we can put them into this Anova table. This is your standard kind of Anova table. So what we have here is the Anova table that we're transferring from that set of calculations. The sums of squares due to row is 3780.75, 3780.75, and there's 1 degree of freedom because the degrees of freedom is 1 minus the number of rows that we have. We have 2 rows minus 1 is 1. Fat type, there are two fat types minus one, so there's 1 degree of freedom, and that sums of squares was 61,204, which we just calculated a few minutes ago. Then we have the row by column interaction, the sex by fat type interaction. That's the degrees of freedom is 2 minus 1 times 2 minus 1, so that's 1 times 1 is 1. And then the error degrees of freedom is the number of rows times the number of columns times n minus 1, the sample size minus 1. And so that would be 2 times 2 times 2, which is 8. And so the degrees of freedom are correct. And that was 11,666.67, which is found right here from the previous page of calculations. Mean squared is just sum squares divided by the degrees of freedom. So these first three things stay the same. The only thing that changes is the sum squares residual, or sum squares within, because it's divided by 8. The degrees of freedom should add up to the correct degrees of freedom, which should be 1 minus the total sample size. And the sums of squares should all add up to the total sums of squares. If you take the mean squared for row, for sex, and divide it by the mean squared error, you should get 2.59. Mean squared of fat type divided by mean squared error, that will give you F of 41.97. And the mean squared of the interaction divided by the mean squared error will give you 0.63. Obviously, 41.97 is a very large F statistic. If you look that up for 1 degree of freedom in the numerator and 8 degrees of freedom in the denominator, you will find that that is the only thing in this ANOVA that is significant. So there's no effect of sex on weight gain, but there is a significant effect of fat type, and there's no interaction term in this particular example. Well, what does that mean? Well, we can graph these data, and I've graphed the means and the 95% confidence intervals for these data, and what you find is that animals, both males and females that are fed fresh fat, grow more, they gain more weight than both males and females that were fed rancid fat. This is that effect of fat type on rat growth. But one of the things that you'll also notice is that males are always higher than females, although not significantly so. So there's no significant difference in the sexes. And because the similar magnitude is the magnitude between males and females for fresh is not that different from the magnitude between males and females in rancid, there's no interaction term because males and females are both responding similarly to fresh and rancid fat. And so that's essentially what this Anova table is telling you. So this is what no interaction looks like. Whenever you plot the data in this way, which is oftentimes called an interaction plot, if you see that these lines are roughly parallel, chances are there is no interaction. So how can we show you what an interaction is? Well, one of the ways that we can show you what an interaction is by basically taking the male data that were fresh and put it in the rancid column, and take the male rancid data and put it in the fresh column, and then recalculating. That's not going to change the row means because all of the data is still staying in the male column, but it will change the column means because the rancid data for males is lower than the fresh data for males, and we're switching them. So it's going to change the column means. So some of the figures are going to change and other figures are going to stay the same. So this is what it looks like. You can see that I've just taken the rancid data and put it in the fresh column, put the fresh data and put it in the rancid column. Now the row means are still the same. Oh, sorry. The total is still the same, which means the grand mean is still the same. So the sums of squares total is still the same. Sums of squares row is still the same because the row means haven't changed. But sums of squares column changes because the column means are now different. You will notice that these column means are now much more similar to the grand mean than they were in the previous iteration of this example. So that when we take 589 minus the grand mean, square that, 606 minus the grand mean and square that, add those together, multiply it times 6, you find that this adds up to 918.75. You'll recall that this is what the interaction term was in the previous manifestation of this example. Sum of the squares within is still calculated the same way. The only thing that has changed is that this mean was fresh before and now it's rancid. This mean was rancid before and now it's fresh. So all that does is it just changes where these things appear in this matrix of sum squares within, but the sum squares within still adds up to the same value. So now you have to calculate the row by column interaction, the interaction between sex and fat type, and you do it the same way. Take the sums of squares total, subtract the row, subtract the column, subtract within, and that should give you 61,204. Take these results and put them into the Anova table, and what you find is that there is no effect of row, there is no effect of column, so there's no effect of sex, there's no effect of fat type, but now the interaction term is significant, and it's the exact same thing as it was before. It's just rearranged because we rearranged the data. So what does this look like when you graph it? Well, how does it differ? Well, it looks just like this. The only thing that has changed is you've interchanged the variation that was due to column is now variation that is due to the column by row interaction term. P-values are all still the same. It's just where that variation occurs in the experiment. Well, what does that look like in terms of graphing the data? Well, this is what the interaction plot looks like now. And it's an interaction plot because fat type is interacting with sex. And what I mean by that is that females, when you give them fresh fat, they grow more than when you give them rancid fat. But males, when you give them fresh fat, they grow less than when you give them rancid fat. And so these lines are crossing, and that is usually indicative of an interaction. And they don't have to cross. They can either diverge or converge on one another. But the main thing is that these two lines are no longer approximately parallel. And that's the visual indication that you have an interaction. Fresh fat affects females in one way and males in another way. Rancid fat affects females in one way and males in another way. And that is the nature of an interaction. So we'll pick up on this idea on Friday, and I will see you in class on Friday. Don't forget to turn in your assignments. I've posted 2 links on the Moodle site for doing that, so find those there in this week's materials. And I'll see you on Friday unless you come by for help with your project.